All right, so briefly, before we dive into today's article, you can find us on neurosci.ro, which is the website. There's links to social media there, Facebook, Twitter, and YouTube, where we post all of our recordings. You can find them on the YouTube channel under the Journal Club playlist. Also, you can access them easier on jc.neurosci.ro. This is the Journal Club page. It's the website that indexes every recording with an abstract and links towards relevant literature. And we're trying to build somewhat of a cumulative series of discussions that eventually, ideally, lead to actual scientific work as well. The previous two meetings were on translational metascience. That was the first one, where we talked about this emerging field that is science behind doing science, which is metascience. And it explores the reasons we have a replication crisis in science. Many results from the literature don't replicate. They're not replicable. And whether they're in smaller journals or larger, higher impact ones doesn't seem to matter. We still have a lot of trouble with replicability of the results we have in the literature all across neuroscience and cognitive science. We're including psychiatry and psychology as well here. And you may remember that the first meeting that we did, we discussed the $20 billion spent by the National Institutes of Mental Health in the United States without ever producing any result that has clinical relevance, which obviously became an issue as the years went on. And we also looked at what Dragos talked about, which was a 2004 article by John Ioannidis on what we call translational medicine, referring to just how little basic research from labs across the world actually makes it into clinical practice. And as you can imagine, research in psychiatry was quite impacted by the lack of translatability from the lab to the clinic, with $20 billion spent without any clinical relevance. And then the second meeting, we actually asked why we read papers at all and whether clinicians should and could reasonably consume scientific literature in order to become more efficient practitioners. And this is certainly an issue when you consider just how much literature is mostly non-translatable waste that will not be clinically relevant. So our point is to try and swing back and forth between basic and clinical research and map out just why so many results that do get published, even in high-impact journals like Science and Nature, will probably never translate to the clinic, and how knowledge can still be advanced with studies that will not necessarily translate to the real world right away. And today's article, which is one such example, because it will probably not translate to the real world anytime soon or to the clinic, was published just a few months ago in June in Nature, so it's certainly a big paper. And it was published by the Arlotta lab at Harvard, which is one of the largest and most powerful labs doing organoid research in the world. As you can see, there's a lot of researchers working in Paula Arlotta's lab at Harvard, and it's a paper that also stems from a series of clinical observations. In pediatric neuropsychiatry, there's a lot of observational data suggesting that prenatal exposure to various neurotoxic triggers has differential consequences for the developmental trajectory of fetuses after they're born. So you have clinical studies suggesting that exposure to ethanol through alcohol consumption during pregnancy leads to a condition known as fetal alcohol spectrum disorder, where you get these characteristic facial features in about 10% of the patients. You get small eye openings, a smooth philtrum, which is the missing indentation of the upper lip, which is thinner. But other than that, patients manifest a variety of symptoms from short height to poor motor coordination, ADHD, learning and speech problems, and even intellectual disability. But we know from clinical studies that not all prenatal exposures to alcohol lead to fetal alcohol spectrum disorder. It's just a risk factor. So you have reviews suggesting that the impact of alcohol exposure is differentiated for developmental outcomes. One of the recurring themes being that the timing of exposure is critical, but also genetic variation in patients possibly plays a big role. Now, however, since we're talking about metascience and replicability, we should acknowledge that a lot of this literature is of low quality and contradictory, so caution is warranted. But there's also preclinical literature, both rodent and primate models, where you can actually do controlled studies in intentionally exposing the fetus to alcohol prenatally. That also suggests that there's differentiated impact in developing fetal alcohol spectrum disorder. So this is one line of observational and experimental research. The other one is in regard to prenatal exposure to valproic acid, which is used to treat manic episodes in bipolar disorder, which is a psychiatric disorder where at least one manic episode has been present and depressive episodes often are present as well. And with manic episodes, you get agitation, distractibility, lack of sleep, excessive energy, racing thoughts, and it's obviously a significantly debilitating condition for which valproic acid is one effective treatment. Despite having fallen out of favor, it's not a first-line treatment anymore. It's been replaced by lithium or other anticonvulsant medications such as carbamazepine, and it's also used to treat seizures, as it's also an anticonvulsant, again, having somewhat fallen out of favor due to side effects on fetuses due to prenatal exposure. It's been linked to autism spectrum disorders. But again, you have literature suggesting that the effects are differentiated. And of course, not all children who are prenatally exposed to valproic acid due to their mothers being treated for bipolar disorder or seizures, not all children will develop autism spectrum disorders. And again, there's animal studies that are better controlled with rodent studies that suggest that there's differentiated impact due to dose-dependent effects, timing of exposure, and genetic variation. So this is the clinical literature that partially generated this line of research. It's clinical studies suggesting there's differentiated impact and developmental outcomes after prenatal exposure to ethanol, with some but not all children developing fetal alcohol spectrum disorders, and valproic acid respectively, with some but again not all children developing autism spectrum disorders. And then there's animal studies for both areas of research suggesting that differentiated impact can be reproduced experimentally and is at least partially driven by genetic variation, which is significantly easier to control in animal studies, particularly in rodent studies. So this is the broader clinical context in which this research we're looking at today emerged. It's going further than just neuroimaging or just animal research, and it attempts to explore a certain special type of organoid, namely chimeroids, in trying to explore this problem of differentiated psychiatric response to exposure to ethanol and valproic acid respectively. And this is what Dragos is going to talk about today, as a lot of his research revolves around organoids.
So I guess, and if I can have like a show of hands, how many of you are first and second year? Okay, so I imagine that not a lot of you have had that much to do with cell culture. So I would like to start by explaining what cell culture is. And first of all, it's a method used to cultivate, propagate, and grow a large amount of cells in a dish. So basically, first we had this pathology like sections that are really, really good at helping us, see what the pathological process is. But they're not that great always at helping us explain it, because usually we take a piece of tissue, we fix it in either formalin or paraffin or whatever, and then basically we kill it. And we catch it in time and we look at the moment where we killed it. So it's not that helpful at showing us, more dynamic processes. So for that, cell culture is a very, very good tool, because we can take these cells, we can give them all the nourishment that they need, and then we can observe them at work. Of course, there are some problems with it, and we'll go into it later. But for you to understand these concepts of organoids, assembloids, chimeroids, I would like to first start by explaining to you different types of cell cultures. And there's a lot of way to classify them. The best way for you to understand, I think, is by starting with adherent versus suspension. So first of all, imagine your blood. Of course, you have cells in your blood, as you might know. So these are cells that excel at living in a fluid environment. They don't really stick to anything, ideally. They move around, you know. So when you grow them in a lab, you want to replicate these conditions. So you would have a suspension of cells in the media. But then there are other cells, like neurons, for example, that need some sort of support. They need to be able to form a matrix. They need that adherence to survive. So for these, we have these adherent cell cultures. And what we do is that basically we take a plate where we coat it with some proteins that will help the neurons adhere to it. And then we put in the cells in a suspension, and they will just fall down and stick by themselves to the bottom of the plate. So what that does, it provides us a thin sheet of cells. So we basically get a 2D culture, all right? Here's where we make this difference between the 2D and 3D. But what we noticed is that if that coating that we did is not really good enough, they start clumping together, because they still need support. So if they can't stick to the plate, they stick to one another. So that's where the idea came. What if we put cells in plates that are very, very low adhesion, so they can't really stick to anything besides themselves? And that's how organoids basically were born. Well, first spheroids, then moving on to organoids and so on. But what are organoids? They're self-organizing 3D cultures. So again, there may be other 3D cultures, like you would have a mold or something, and you put cells on it and they grow, you know, like, I don't know, imagine basically doing a bone, a 3D printed matrix of a bone, and then you grow cells on it and they grow. That's still 3D culture, but that's not an organoid because we organized it. It's not self-organizing. So we just leave the cells, they organize by themselves, and they should resemble either the structure or the function of an organ. So that's why they're organoids, all right? The problem with them is that we start from stem cells, and we need to we need to differentiate them. So what we do is that, first of all, we get them to germ layer, and then from germ layer, one step at a time, to differentiated cells. So we're very restricted in the sense that we can get a maximum of one germ layer. So I'm not sure how many of you have done this in histology, but do you know around what kind of cells do you have in the nervous tissue? Anyone can, if anyone can. Aline, can you help me with that? What types of, what types in nervous tissue?
Like neurons
and glial, yes, cells. And for glial, we usually have three main glial ones. In the central nervous system, we have oligodendrocytes, astroglia, and the last one? Microglia. Now, this is the problem with the microglia, because they're basically the immune cells of the brain, and they don't really form in the same germ layer as the brain. They actually come from like blood cells, and they migrate inside the brain. So when we do these organoids, we will get all the cells, almost all the cells in the brain, but we will not get the microglia. And then there came assembloids, where we either combine two different kinds of organoids, or we just add another type of cell in it, which is like the microglia. So how assembloids work, imagine we do a brain organoid, a spine organoid, and a muscle organoid. We put them together, they combine, and then they transfer the information from the brain to the muscle, and we will see the muscle contract. That's one of the more famous ones, but there are a lot of ways of doing it. So basically, how exactly do we make these organoids? First, we start by making a 2D culture of stem cells. Those iPSCs means induced pluripotent stem cells, which means that usually we will take like a bit of skin from a patient. We take the fibroblasts in the skin, we transform them to stem cells, and from stem cells we can transform them to whatever we like. So we do a 2D culture of stem cells, then we put them in those low attachment plates that I told you about, and then they form these embryoid bodies. We start neuroectoderm induction. So basically we start step by step differentiating them, and then in the end we get the brain organoids. There are two main types. It's either cerebral, which has all the regions in the brain, or region-specific ones. It depends what we want out of it. If we only want to look at the cortex, then we can only do that. If we only like to look at, like the substantia nigra, we can also do that. But it depends, and if you want to look at whole brain, the cerebral ones are better. So there are uses, and I won't get too much into those. For assembloids, again, I won't get too much into those. It's, as I said, you either combine more organoids, which is the multi-region assembloids, or you put, let's say, microglia in an organoid, and that's multi-lineage assembloids. They are a very, very good model because they provide us more complexity than a normal 2D cell culture would. And what we see is that a lot of times in 2D cultures, we see that a lot of treatments work wonderfully. And then we take them, we give them to patients, they do nothing, or they even make them worse. It's because we lack complexity, we lack tissue heterogeneity, and this model actually allows us to get that heterogeneity. We get different types of cells into one culture. When we have 2D cultures, we need to constantly keep moving them from, it's called subculturing, but the idea is that in the end, you lose different kinds of cells, you get a homogeneous culture. Here we can get a heterogeneous culture, and that's very, very good. These are some pictures at different stages from day 0 to day 28 one, which you can see it looks kind of funky. And there are multiple uses for this. I won't get too much into this because I want to get at the actual article that we're talking about. But we can discuss them more if you want in the discussions part. There are a few limitations, and of course, besides the fact that, again, it's in a culture, it's in a lab, so we can't really mimic the body perfectly. We can't mimic all the conditions that we want. If you remember the steps I told you, we start by taking stem cells. Those stem cells come from a piece of skin from a patient. But that means that even if I do 50 organoids in a study, I'm actually studying the same patient 50 times. And then I'm trying to take, to make conclusions out of that and to extrapolate that to the general population. And I think you might see why that's wrong. But to make it more clearly, we can get false positives. We can't underestimate the variability that we get, because of course, if you only see variances in organoids that come from one person, you may miss a lot of other variances that may come from a lot of patients. They're not that representative because, again, it's a one patient study basically, and there are problems regarding the reproducibility, because if I see, okay, so they did this study on organoids, I want to reproduce it, it's very little chances that I will get the exact same stem cells that they did from the exact same patient. So then I'm doing my study with another patient that has different genetic makeup. So in the end, I may have problems. I may not be able to get the same results. So then it's a problem of reproducibility, reproductibility. And so is that really science? If it's just a finding that we found, but no one else can reproduce it? And then it's, since it's so like one patient, it's very easy for us to misrepresent diseases. You know, we may not understand it, and we may think that this is the way it looks, but maybe this is the way it looks just in this patient. And then, again, as you might know, at some point there may be mutations in cells. So for these stem cells, if, you know, like I keep like culturing them, if I get a mutation at some point, I may just like keep on cloning the same mutation that's not actually found in the patient that I had in the beginning, you know. So this is a clonal artifact there. So basically we have two solutions. One of them would be to make, in the same conditions, a lot of organoids from a lot of different patients. But the problem with that is that it's very expensive, first of all, and second of all, you still, if it's the different genetic makeups, they may have different metabolisms, they may alter each media that we put in. So we're not sure if they are actually growing in the same conditions. And that may also provide some problems for us, because it's not that well of a controlled environment in the end, you know. So maybe you know what a chimera is. It's like this monster that has like a lot of different animals stuck into one. But right now it's a word that's used for basically any hybrid creature. In medicine, chimerism is when you have like different DNAs in the same patient. So that's how they went with like a chimera organoid, a chimeroid. Because what they actually do is that, again, if we go back to the steps of making an organoid, what they did is that they took stem cells from different patients, and instead of making a lot of different organoids, they put them together to form just one organoid. And remember, we are limited to one germ layer cell type of cells, so we don't have immune cells here, so there's nothing to attack, you know, like other cells. So they can live just fine together. And it's pretty interesting. But what they did is that, okay, so they put in cells from different, pluripotent stem cells from different patients together, and they let them grow into those embryoid bodies that I told you about, you know, like this, before we start differentiating them. But what they notice is that, well, some of the stem cells are much more proliferative. They divide much more. They're better at fighting for resources, while others aren't. So what they got in the end is, and if you can see here, is that basically mix 3 and mix 4, they did like 5 mixes, that I won't get too much into that, but 2 of their trials ended up with having just basically one line surviving, and all the others dying because they couldn't access resources, you know. So it was this GM line that they have, and all the others were dying. And even in mix 1, where they still managed to keep some heterogeneity there, it's still like 85% of the mito 210. So it wasn't ideal, because again, it kind of defeats the whole point. If you never know whether the cells that you get from a patient are more proliferative or not, and if you don't know how much of each cell will be represented there, it defeats the purpose. It's not reproducible anymore. It's so it's a big problem. So what they did is they thought, okay, what if I let them differentiate more, and then I do the chimeroid thing. So they did that, and they let them become from pluripotent stem cells to neural stem cells. And those divide much less, and what they saw is that they actually also live together much more peacefully. And you can see that basically in each batch it's a much more similar distribution, and they managed to keep that diversity. And basically all the cell lines that they put in also managed to make all the types of cells that you would see in like in a human brain, except like the microglia. So how did they make this? We go again back at the protocol for the organoids. We take stem cells from one patient, but we do it with like all the lines in parallel. We put them together in cultures that are low attachment plates. They become these embryoid bodies. We start slowly the differentiation, and what we do then is that we take these embryoid bodies, we break them up, we make another single cell suspension, and then we mix them together equally, and then we put them back in so that they form the embryoid bodies again. They reform it. You can see here the reassembly period. So we break them up, mix them up, and then we let them reassemble. And then, only then, we start to differentiate them. And what they saw, again, is they got much, much nicer batches. That it's interesting to me, and I will get more into that when we discuss it, because usually cells are very, very sensitive to having their ties, like their connections be broken up and then having to restart, and a lot of them die. So I'm very curious how many tries they had to do before they got it. But it seems like, and they said that it's pretty reproducible. And here are some pictures. And probably most of you have not seen immunocytochemistry pictures before. But what we do here is that we get an antibody, we put it on whatever you want to look at, specific proteins or genes or so on. And then we get another antibody that is against the first one, and that also has a molecule that glows. So that's how we get different colors here. So here, the blue one, the DAPI, is something that stains whatever nuclei we have, so that it just helps us make sure that they're alive. It's a reference, we know where the cells are, that's not as important. SOX2 goes to progenitors. So we have these rosettes that we would expect to also have in normal organoids that are progenitor cells. So from there you can imagine that they grow, and from there we start getting these more mature cells, and that's how they grow. And you can see that here it's more connected, and then they start getting some space in between them. Then MAP2 looks at dendrites. So that's how we know that we start having more mature neurons, and also TBR1 is for deep layer neurons. So we also see that there are some more mature parts, and also some parts that are still young and still proliferate. And that's the same here, but with Z01, which is like tight junctions, and EMX1, which is for dorsal cortical progenitors. It's not very important. I wanted more for you to understand that they look like normal organoids, and also a bit about the immunocytochemistry process. And at this point, to be honest, I'm just putting up pretty pictures. But yeah, I really enjoy them. But then what they did is they needed a control, because they can't really use normal organoids as control, because the whole process of creating them differs. So they did this single donor neural stem cell chimeroids, which why I put them in inverted commas is because they're not actually chimeroids, because they're single donor. They're basically organoids, but they let them become embryoid bodies, they broke them up, they let them come back together. Again, just one donor, comparing to the others, which are like the multi-donor that have more. but they need to go through the same process so that we know that it's not the breaking apart and putting them back in that actually changes stuff, if there are any changes. So what they did is they basically compared this multi-donor with the single donor one with neural stem cells and also neural progenitor cells, which were a bit more differentiated, and they saw that it's better. Don't mind that too much. But what you should see, and they also compared this with data that they got from actual fetal brains. So what you can see here is that basically the distribution is pretty similar. And among them, the images look pretty similar. And that tells us that it's basically a good model, and that the fact that we have more donors doesn't actually impede us, because the single ones and also the fetal brains basically look very similar as well. So it theoretically should not be a problem that we have more donors. So now that they established that this is a good protocol, as they wanted to actually give us a way to try it, like a use for it. And here, don't really take this as the main part of the article, because the main part was them showing up chimeroids. But this is a proposed way to use them. And what they did is that they treated them with ethanol and valproic acid. And here I will go into this in a bit, but I want to help you understand the protocol, because I'm not sure how many of you read protocols before, like cell culture protocols at least. So here you can see that we start the experiment when they are day in vitro. So that means that we started the culture 45 days ago, 45 days before we start the experiment, we start the culture and everything. So that's when you would normally start this differentiation period. And it's very important because here they look at ethanol. So it would be like, like the alcohol baby syndrome with mothers that drink during pregnancy, and also mothers that take the valproic acid during pregnancy. And it affects them mostly when they're still differentiating. But what do we know about ethanol, alcohol in a room? What does it do usually if you just leave it there? It evaporates, exactly. So especially here in these conditions, we keep the cells at 37 degrees to mimic the temperature of the body. We have evaporation. So they know that basically the alcohol would evaporate from the cell culture in 2 days. So every 2 days, they put in more alcohol, so that they keep basically a stable line, a stable amount. And also they took the amount for both the ethanol and the valproic acid from actual clinical studies, so they would get the same concentration. They don't bump it more than you would find in an actual living mother and its fetus. So they did that, and then at 15 days later, at 30 days later, at day in vitro 75, they stop it, because that's basically at maturity when the baby would be given birth. And ideally the mother would stop giving the baby alcohol once it's been given birth to, So here they stop, they let them mature, and then they do the sequencing. And then for the valproic acid, it's almost the same. But the thing is that the valproic acid doesn't evaporate. On the other hand, it does. A part of it stays in the media, a part of it breaks down by itself just because it's in warm conditions, and cells tend to, substances tend to react with stuff when they're in, especially when they're in warm conditions. And then there are also cells that break them down and use them. So basically, at some point you lose you lose a lot of valproic acid, you lose part of the concentration, you get a lot of byproducts like metabolites, toxins, whatever. So what would you do? Would you just add more? Ideally no, because you don't know how much is left of everything. And if you add more and more and more, you then get a very, very toxic environment with a high concentration. So what they did is that they changed the whole media. They took the media out, threw it out, they put in new media, and then they gave it again the same amount of valproic acid every 3 days. So that's how they kept a stable amount, a stable line. And then again, they let them mature and so on. After they did both these experiments, part with alcohol, part with valproic acid, what they did is that they basically looked at how many of each cells were still alive. And now this is where I have a bit of a problem with this, because toxicity is much more than cells dying or not. You know, there are a lot of states that the cell can be in, and toxicity can mean a lot of things. But here they looked just at how many survived, and you can see like complete, like in total, you had like 103,900 whatever cells, and then for ethanol 60,000, and then 118 and 11,000 cells for valproic acid. And then they look for each of the lines, and then they see that some lines more died, so that means that it's more toxic for these, so that genetic background probably has some sort of like sensibility for this for alcohol or valproic acid, and some survived, so maybe they have some protection. But I think that's a bit reductive to what toxicity means. So here is my opinion, so don't take it with a grain of salt, but I think that they did not really achieve what they said in the title. They didn't look at individual susceptibility. Because something that you have to understand is that for us to look at cells, we can usually, if we want to test different like molecules that are in there, if we want to test here it was DNA, it was sequencing and so on, we need to kill them, we need to break them apart, and we need to look at the parts. But if you do that with a lot of cells, they get mixed up. So that's why you look at very specific things. Here they looked at genes that they knew were in each one. So that's how they differentiated how many of each cell were there. But at the same time, you cannot look at both what type of cell it is and what proteins they actually did only in total, but you can't really see, okay, these cells secreted this metabolites, this, you know, because it's just, you can't do it in with the same cell that you destroy. So you need to pick one, to pick your poison, to pick whatever you want to look at. In normal organoids, you wouldn't care about that because it's just one line, so you already know the genetic background of it. You know, so you only look at whatever the effect was. But here you need to know both the genetic makeup and the effect. So basically the easiest effect you can look at is cell death. And again, there you have to extrapolate a lot of things. It's not the best way. But I think that it's a very, very good model for something else, and they also mention it in the discussions, is that the chimeroid system makes it possible to investigate in one single organoid the collective development and behavior of a larger diversity of cell types of the brain across multiple human individuals. So it's not a very good, in my opinion, a very good model to look at individual changes in each of the individuals, but it's a very good model to look at populational changes. Because unlike in the normal organoid where you have one individual, here you can put basically in as many as you like, not too many because it's very expensive and because you start to lose your way of like actually recognizing them. But you get more, you have something that's more statistically representative, and that's where I see the actual like benefit of it, you know, in helping us reduce selection bias, basically, not in looking at individual changes. For individual changes, I think still the normal organoids are better because you can actually look more in depth at those changes. And now for discussion and some conclusions, it's basically one of the closest in vitro models to reality. Organoids and assembloids in general, but chimeroids here as well. it's very, very expensive. Even more so than organoids, which are usually an expensive model, because here you need to do sequencing every time, which is a very expensive procedure, and you usually don't really need to do it as often with other models. It may help, it helps with the lower reproducibility of normal organoids, but still accessibility is kind of a problem here. And they still really can't mimic the exact environment that you would get there. So there's a lot of room for innovation here. And yeah, thank you. And I hope that it wasn't way too confusing for you, but hopefully now with the discussion part we'll be able to get more into everything. Okay. Questions, comments?
I would like to know in more detail what exactly happens in the differentiation period. Like, what do you do in the lab?
Yeah, that's a very good question. So, and I hope it may be a bit disappointing. It's not like that very, very impressive. But what we do is what also happens in the brain when it develops. Basically, you have these chemicals that tell each cell what to do and what to become. And we know what these are. We have variants of them accessible to us, so we just give the right chemical, be it protein, be it whatever, vitamin, and so on, at the right time, which was studied more and more to see until we get to the point where we want them to be. And even then, we actually call the cells that we differentiate, we don't call them neurons, we call them neuron-like cells, or microglia-like cells, because they look and they behave like neurons, but we're not sure that it's perfectly the same thing. Okay, other questions? Why it's a good model, and that reminds me, because I had a talk with Aline at some point about doing primary cultures where you get the cells directly from a patient. In neuro stuff, you can't really do that, because I can't go in and take healthy tissue from the brain of a patient and put it in culture, because I would give that patient different disabilities. Whereas this, I can take some skin and I can transform it to brain tissue. So that's a very, very good, helpful way of studying brain tissue, live brain tissue. Yes?
So why can't we mimic tumor?
Why can't we, what?
Mimic tumor.
Yeah, that's more organoids in general, not these chimeroids, but I will go into that. The thing is, first of all, we don't have all the cells that we have there, because what we see here is that tumors make this microenvironment, and they get the immune system to also help defend them, and that's something that we can't really get here. And then we also don't have we have different, the media is not the same thing as the cerebrospinal fluid, for example, So, and it doesn't have those conditions, it doesn't have those cells attacking or defending or so on. So what happens is that cancer is something that keeps mutating, keeps making as many copies and different mutations. It's not just one cancer. You actually usually have multiple type of cells and mutations in the same tumor. So basically what we do here is that we give them a very easy environment to live in. And in reality, it's not as easy, for them. So basically we can get mutations that would never survive in an actual living person. And maybe some of the ones who would survive get out-competed for resources, and then we don't get them. So that's basically the gist of it.
All right, so I wanted to actually give my critique of the study, and I'm gonna start by addressing the fact that I don't think that in the study the actual population was detailed enough, and the actual selection of the cells and the donors was based more of like on a gene thing, which at least that's what I understood. Like they basically selected for specific gene variations. And for me, I think even if we get to, use this technique in the future, I think there needs to be more of like, you need to basically account for more gene interactions, if you know what I'm saying. Like, you can't just look at a specific gene variation. You have to look at like the whole thing and to try and find the most similar like phenotype of cell that only has that specific variation in the gene. And I think that's very, very hard to get. Like, I think it's almost like quite impossible.
So to your point, I think that that's not as much of an important thing there, because usually here again, if you want to look at how multiple individuals work, if it's ideally, like a randomized control studies or so on, you would need to get something that's more representative of the general population. So I wouldn't worry that much that it has this specific mutation or so on. I would rather just I think they just took whatever lines they had and then they looked at the genetic makeup so that they can recognize them later. But I don't think they selected them based on the...
Yes, but that's what I'm saying. Like, if you are to implement this technique, like further on, I think that should be like an improvement we should do,
Why?
Because I think that'd be a more representative thing, more representative study.
In a way, well, of course, it always helps us to have more information, I guess. But at some point it becomes, I think, a bit... it may be a useless cost, in the sense that it's a lot of work to do sequencing and to look at every, genetic mutation and everything. And at the same time, I don't see exactly what more you gain. Because... again, you want basically more patients in. So this is a way to get more patients in rather than just one. Again, if you want to look at a specific mutation, let's say that it's a disease that's very, very, like leaf-manures, whatever, then you take that mutation, but again, you take it from different patients who may have other mutations as well. But you don't care because that's how you make it more representative, because you get more patients in,
Yeah, I know what you're saying, but I'm saying like
it takes from the blindness of the thing.
I know, but like the conclusion it basically had, and even the title is kind of clickbait, because it kind of makes like a clinical conclusion.
Yeah.
Out of like a fundamental study, which is like not, you shouldn't do that,
Yeah, I agree with that. Clickbaity. Maybe it's a bit clickbaity. I don't think that they actually achieved what they said in the study, because I think toxicity is much more than just like cell death. But I think that that's just again, the purpose of the study in itself was more to be
bring awareness to
this model. It's a very cool model. Here's an example of how you can use it. But I kind of see what you mean, but with just that idea that it may be a bit redundant to go too much and spend so many resources on just making sure that you know everything about that.
Yeah, I'm saying I would rather them just don't make such conclusions, out of these kind of studies. Just it should be just a thing that we you don't make clinical conclusion out of it. You just try to find new mechanisms, and then you do the whole research thing, the whole research.
Yeah, again, especially because I don't think that the study was for this.
Yeah, it was just a thing.
It was such a small picture of what was actually presented. If you look for it, it's just one chapter out of everything. I agree. But at the same time, I also get that they wanted to, show off their model in some way.
Yeah.
And it's more appealing that way.
And also I wanted to, like, in the same sense, I wanted to add that, like, it's good that they, like, study the fact that, you know, like, those, like, different cell types can live with each other, you know, and it's kind of like similar to a normal human brain. Like, they don't influence their development, but my problem is they may influence their different metabolisms, you know. Like, maybe one cell type makes a certain metabolite which influences the other cell type, and that's why I think you can't make, like, this kind of, like, conclusions that this cell is more like this cell alcohol is more toxic to this cell, because maybe that cell is influenced by the other cell, you know.
Yeah.
Stuff like that.
No, I get what you mean, but I think that for that it's very important to have a lot of controls, you know,
Yeah.
and to make sure that because a lot of times you'll see that it's better to have way more controls than it is to have, you know, like that many of your studied population, because it's easier to do the comparisons that way, I think, and it's easier to make sure that you don't get like these false positives there. But yeah, I really agree with you there. Yeah.
And also, I think due to the fact that it's a very complex protocol, it takes a long time, and, you know, even the making of these chimeroids takes a long time. I think it's very hard to reproduce, you know. Even taking out of, like, even if you have the infinite amount of money, I think that, like, for example, you make such a little adjustment, like, at the beginning of the process, and because it takes such a long time, maybe at the end you get, out of that small variance, you get a very, very big difference between studies. And I think that's, like, why these kind of studies aren't that reproducible.
Yeah, that is true, but I think that, again, with infinite resources you can afford to make many more batches, you know, because you won't actually take conclusions of just one batch. That's way too little, so that you can try to get, like, these small variances out. And again, what this actually helps is that, again, you don't get because your critique is also fair for organoids. Organoids also take 3 months, for example, or more, up to 1 year a lot of times in culture. And here you actually have a way to test it, but on multiple patients, not on one, you know, so it actually helps reproducibility there rather than impedes it that way. But yeah, that's a more of a...
Like a whole organoid, yeah.
Yes.
And that was basically my 2 cents about, like, why I think this study is kind of like, you know, the reproductive. Well, I thought I talked about the reproducibility and the representativity of it, you know.
Mm-hmm.
And, you know, I just wanted to, like, basically, like, my whole, in conclusion, I think this study is very, very, it's a very good study, and it kind of brings into front view a new technique, which I think should be used a lot more. But that the main problem, I think, was, you know, they tried to like say conclusions that weren't, you know.
Yeah, the conclusions part as well, but what I think, again, where I see them making a bit of a maybe bigger mistake, but again, this is like the Aglota lab, which is like very big, so who am I to you know, critique her? But I think that they're actually focusing on the wrong thing here, as I said. Trying to look at individual changes may sound more, you know, like interesting, may sound more appealing, but at the same time, I think it's much simpler than that. I think it's just a better model, usually, you know, like, if you have enough resources to do the whole sequencing, to get more cells and so on, I think it's better to do your experiments on this model rather than normal organoids, just because it's not an n equals 1 kind of study. It's not a one patient study. But again, this is actually a very, very big pro of it, but it's not as, you know, like, shiny as being able to look at each individual changes between them or whatever.
Do you think it would have been better to just do, like, more organoids than just a chimeroid?
No, that's what I mean, is because here you can make sure that all these patients are in the same environment. Whereas when you do a lot of different organoids, you get different batches, and as much as you try, you won't get the same in the same conditions.
So you're saying there's like no, there's not been a way discovered to basically make the.
Because you can't control how, as you said here, as you saw here, it's different proliferation rates as well, and the metabolism of each like individual based on their genetic makeup is different. So you can't really control where that media that you put in ends up and what it does, But here you can make sure that it's in the same conditions, because you have around the same amount of cells from each patient living together. So I think it's better in that way.
Mm-hmm. I get what you're saying, but like, I think, yeah, as you said at the end, I think it's better to just see like the development of different cell types, as you said, not introduce an external factor.
Wait, what do you? No, I think that you can introduce external factors here, but in the way that you study it, you don't study how it affects each of them.
But like the whole.
It affects the whole.
Yeah. Wouldn't you think that a model like this would be better for example, to study the effects of alcohol on different like neural cell types, for example, from the same patient? You get what I'm saying?
Yeah, but then if it's from the same patient, why do you need the chimeroid?
You could like make an organoid which represents every single.
Yeah, but then you don't need the organoid. If you want to look at them differently, you just make like 3 or 4 different 2D cell cultures of each of them. If you get the benefit of the organoid is that you get them together.
Yeah, that's what I'm saying. Like to make like a micro brain, for example.
We're moving away from that because it's not actual mini brains, but yeah, basically that's what an organoid is. So
yeah,
at the same time, it's just the normal organoid, and you can look at whatever cell interests you. Then,
okay.
But that's if you want at one specific patient, then you don't need the chimeroids. If you want in general, then again the chimeroids are better because you get oligodendroglia from all these patients, neurons from all these patients. So you can get a better guess of what it would like in general.
Okay.
Yeah, it definitely is a lot of trouble with translation, and these again, of course, if you get, and we discussed that you can actually get the journal club recording of the session where we discussed translatability in science, and I especially discussed a lot, I think, about organoids there as well, is that when you have cell culture, of course it's very different than what you get in the actual thing, because there's an entire system, very complex system. Here you have very, very controlled conditions where you try to put in as few things as you can so that you can, be certain that the effects you see are in those things, and you try to control everything as much as you can. So of course then you try to take the assumptions you got from there into actual practice where you have a infinitely more complex system. They don't translate that well. But organoids and I think chimeroids will also be there, but organoids and assembloids, which are a bit more well defined right now, are the best cell culture, like in vitro way of actually predicting what happens in the brain, because it's the most complex thing right now we have.
What did you guys think of the study? Have you guys read it before?
Can I get a show of hands who read it before? Okay.
Was it too difficult to read, do you think?
It was a bit difficult, and I find it really interesting, but I'm still wondering how useful it really is, like applicable in medicine, because to me it's not clear how you get to the root and the cause of the susceptibility to toxins in different genetics.
Yeah.
Because it showed us really well how different type of cells react to these toxins, but it's not clear to me what made some cells stronger, let's say, than others.
I agree, because this...
The gene expression for different cell types from different donors.
Yeah, but I agree with you, it doesn't.
Yeah.
And it's not supposed to. Here what it's supposed to do is that you see these differences and you know that, okay, these seem to be, more susceptible, these seem to be less susceptible. Or you see that, let's go beyond that, you see that there is an effect here. So you know that, okay, something in this actually holds the answer. Because otherwise, how do you know that it's not a blood thing that causes it? You know, how do you know that it's not a, toxin thing that you got in or whatever? You know, so here you can get a more controlled way. And let's take the ethanol and the susceptibility. Let's say that I agree that the cell death thing that they did means that looks at exactly at toxicity, you know. Let's pretend that there is nothing more toxicity than that. Then I know that in this line, a lot of cells died, and in this one, they actually were able to resist. So somewhere in the differences between these genetic makeups, it's most probably the key. There is most probably the key to what made that, what made them behave differently. So that actually informs us for a further study, you know. Yes.
Like a pre-study for
Yeah.
further detailed study.
Exactly. Because you need to think, before we get to an effect or a phenotype, we start from DNA. We then go to RNA. So if anything goes wrong in that part, something like, first there can be a problem in the DNA, right? Okay, but what if the DNA is perfect? Then there can be a problem in the part where you transition from DNA to RNA, you know? Then there can be a problem where you transition from RNA to proteins. Then there can be a problem in the function of those proteins and whatever like enzymes you get in the end or whatever. And then it can just be a multifactorial thing. But anywhere there, something can go wrong and you can get a disease. So it's very hard to, you can't really know without these sort of studies where to look. Yeah, so that's why we're trying to take something more complex, very, very complex, and then dumb it down as much as we can to be able to find a niche. Basically, and in the end, the answer. Did it make sense or? Okay. But I wonder, out of those of you who read it, how many of you got a good understanding of what it meant? Just from you reading it.
It makes much more sense now after the presentation.
Okay, I see. Yeah, because I know that they can be a bit, scary. A lot of new information and terminology there. Yes. And especially if you don't have the background of, cell cultures first, 2D cell cultures, then 3D, then organoids, and so on, to get to this point, which is the cutting edge of organoid, then of course it's going to be harder to understand.
Yeah, and also they probably haven't gone through MCS and they haven't learned how to actually, how research is approached, how to actually read a study. That's kind of a very difficult part for, most people at the beginning.
I am recorded, so I will not go into what the people who do go for MCS I left you afterwards, but yes, I get what you mean.
Yeah.
Okay. But what I would like, you to get afterwards, because this is a very technical one, so probably very few of you, if any, will work with chimeroids. But you can see that there is this very cutting edge model, but you can also see that there are flaws in it. And the thing is, whatever model you do use, you will never have a perfect model. You'll never have something that completely mimics the human body, and if you do it on the human body, like on patients, you will have no real idea if what you did actually caused it or it's something else, because it's too complex of a system. So there are always pluses and minuses, and it's very important that you're aware of these limitations when you choose what method you want to use. Okay, and this is more like to illustrate and for you to realize, because I think that, again, it's a Nature paper. It's a very fancy sounding thing, it's a very expensive thing, and you usually would think that, okay, I can trust these results, you know, and even these models have big problems. So again, that is for you to, first of all, be able to always question results. You know, we also had a talk, and I think we also mentioned in the translation problem journal club, that there is this guy who retweets headlines. You know, like there was recently a headline which we might have found that there is this immunotherapy that's very good at curing alopecia, you know, baldness, and it was done in mice, you know, so that all that Twitter account does is that it retweets it and it says in mice, you know, because then it very rarely actually gets to translate in people. It's the same here. It would be in organoids, you know, or in cell culture and so on.
Always, like, I think like the main conclusion would be to always like take things with a grain of salt when you read a headline.
Yes, but then also don't get into the very pessimistic part where you're like, okay, so we don't do science anymore because no models are perfect and we can't really know anything for sure anyway. Because again, there are also a lot of benefits to these methods, and we can get quite some information from them, and it may help us change clinical practice as well, if we do it knowing what the limitation and everything and being cautious about them. Okay, so does anyone else have any comment or question or anything?
So I read on the presentation something about personalized treatments, like we would be able in the future to use those chimeroids studies for personalized treatments for patients, right?
So, yeah.
If we use them effectively, the chimeroids, and we study more about different treatments or toxins, and we see how, like the title said, individual susceptibility, but I'm wondering how realistic it is given the price.
Yeah, so a quick disclosure. I had a previous presentation on organoids, and I also used a bit of some slides, and that was more of an organoid thing rather than a chimeroid, because in personalized medicine you actually care about that one patient.
Yeah.
So chimeroids don't really help with that, because
Let's say we can just take skin cells from that patient and
yes,
if we can do this,
but it's with organoids.
Yes, we can find that.
And the idea is, let's say you have a multiple sclerosis patient, right? And right now, multiple sclerosis patient will get the diagnosis usually already a bit late, and they will have to go through different monoclonal antibodies until they find one that works for them. And even that, you don't know about like the toxicity, you don't know how much it will take and everything. So ideally, with the more these methods are used, they become cheaper. You would be able to, like take some skin, make a bunch of organoids of that patient, treat each of them and see how they each respond to the, like basically try it on the patient, each of the treatments, and then see to which they respond best, to start with that one. You don't have to put the patient through months of just adverse effects with no benefit.
So you do it on the patient, but not exactly on the patient.
Yes, exactly. And you can do it on multiple versions of that patient, basically, because you can try a lot of different treatments at the same time, see which it works, so you start it as soon as possible with the real one, on the real patient. That's the promise of personalized medicine, but again, that's more organoid, not chimeroid, because again, it would be counterintuitive to do it on a chimeroid. Okay. Anything else? If not, I would like to thank you very much for being here, and I am very, very happy to see that you did have quite some questions, and you did get, like a lot of the problems that were with this, and as you can see, with the cost as well. That's another one of these, and I really hope that we're starting to get more and more used to reading these studies, even if they get a bit harder, and try to look at them as critically as you can. So yes, from this I really hope that you at least got more of a idea of like how different methods may work, what some flaws you can find with them, some problems that may arise in different methods actually, with reproducibility, reproductibility, replicability, with translation. And I really hope to see you next time. We'll have a more Christmassy edition, and it's going to be a study on fMRI and finding this Christmas spirit on it. It's going to be quite fun, I think. So yeah, thanks again, and see you next time.
Thank you.